Hello, everybody, and welcome back. My name is Dr. Dan Stern Cardinal. You may be a little bit confused seeing me because you haven't seen me yet this semester. The reason for that is that my wife and I have been home with as of the day you're watching this, probably, a 10-week-old baby who was born in August. So I've been home on family leave. But now I am back, and I will be with you for the rest of the semester. I am the normal general biology instructor along with Dr. Beal, but I've been out. So the first thing I want to say is a giant thank you to Dan for covering for me for all this time. I'm sure all of you love Dan and are going to be sad that he is leaving. A consolation prize is that my name is also Dan and I also have a beard. So if that helps soften the blow of changing instructors mid-semester, I hope that helps a little bit. Yeah, let me just quickly introduce myself actually. So this will be my, I don't even know what year, this is sixth year doing general biology, being a general biology instructor here at Rutgers. And before that, I was a graduate student here at Rutgers, and I was a TA I was a workshop TA for general biology. So I've been doing this for now 6 years. And for 6 years before that, I was a TA. So I've been involved with the general biology program here for a long time. I'm thrilled to be here. The woohoo is not part of the title. That's just me because I love genetics and evolution. And so I get to jump back in right into the stuff. That's kind of my favorite part of biology. Although, as you will see, I will say this is my favorite thing a bunch as we go to the point where you will probably laugh at it a little bit each time I say it. So, just a quick note before we actually get into today's stuff. I'm going to do this straight through in one take, basically. We'll do it live, as it were, for those of you that are old like me. If you don't know what that is, just look up, we'll do it live, you'll see. So there's not going to be like fancy editing. There might be pauses, or I might have to go back or something if I say something wrong and then catch it. I apologize. But with so my wife and I have the baby, and we also have a 2.5-year-old. So things are very busy here. So in terms of just the amount of time I'm going to spend editing these, it's not going to be as much perhaps as you are used to. And for that, I apologize. But that's just kind of the logistical constraints right at this point in time. So just keep that in mind as we go. We're going to just do this straight through. And yeah, I hope you like it. And so just as far as review sessions go, Dan is going to be with you through the material that he did. So he'll be with you on the day you're watching this, on Tuesday. And then, or I should say the day this becomes available. And then once we kind of cycle through, then I'll pick up. So on the following Thursday, then I will be also present at the review sessions. We'll swap at that point. And that's the plan. So here we go. Today we are talking about genetics, which is one of my absolute favorite topics. I'm so excited for this unit. So today we're going to kind of just introduce the basic concepts. We're going to start by talking about Gregor Mendel and his experiments. So the outcomes for this section are to sequence Mendel's work, compare and contrast the two competing hypotheses that we're going to talk about, and compare and contrast the principles of heredity that Mendel developed. Then we're going to look at genetic crosses, what those are and how they work. So sequence, compare and contrast, and apply genetic crosses. We'll look at kind of different aspects of genetic crosses. You should be able to kind of go through the process and use the tools that we talk about. And then finally, we will look at how probability plays a role in genetics. So be able to apply the probability rules that we discuss. And this is going to be really important because on the exams, you're going to have to answer questions about genetics, about the outcomes of genetic crosses. And in doing that, you're going to have to do calculations, not complicated calculations, but doing them with the probability, doing the math is faster than drawing out Punnett squares each time. So strongly recommend you practice with those rules. Now, before we get into the material here, we do need to just cover a little bit of background. Now, the basic idea underlying genetics, that kids resemble their parents, that's nothing new. You go back centuries or even millennia. Think about like the first agricultural revolution about 12-ish thousand years ago, 15,000 years ago even, when like domestication became a thing. We knew that offspring resembled their parents, and you could select for certain traits that you want in those offspring. Now, the mechanism of that inheritance was unknown until the 20th century, and as we go through this unit, we'll get to how we figured that out. Charles Darwin, who we're going to talk about later, you may have heard of him before, he's, kind of important, came up with this idea called evolution by natural selection. Now, he didn't know about genes and chromosomes, and he published his work in 1859. He didn't know about kind of the underlying cell bio of any of this, and that's also true for Gregor Mendel, who we're going to talk about today. But what both Darwin and Mendel did was make observations and then come up with the kind of underlying rules to explain those observations, and that's what we're going to talk about here. Before we get into Mendel's work, we do need to cover a little bit of vocabulary. So the first word I want you to know is gene. Pretty simple. A gene is just a unit of hereditary information. So we all have genes, we've inherited our genes from our parents, and the genes are kind of the units within our genomes. And if we're being a little more technical, which we're going to get to in a little bit, genes are the things that code for proteins in our genome. Now, allele refers to alternative versions of genes. So for example, if you have a gene for hair color, you could have the allele for blonde hair or black hair or red hair. That's what we mean by allele. It's a variant of a gene. Now, kind of speaking a little more generally, we also talk about characters. Character just refers to observable, heritable features of organisms. So like hair color is a character. And then trait or character state refers to a detectable variant of that character. So whereas hair color would be a character, red hair would be the trait or character state. And then finally, we have two words you may have heard before, genotype and what's the next one? Yeah, it's phenotype. So genotype refers to the genetic makeup of an organism, in other words, what alleles are present. And the phenotype refers to the observable physical traits of that organism. And you can see that these terms are kind of related thematically to each other. There's differences, for example, between character and gene, but those two things kind of go together. So you can kind of look at it like this. Gene is the part of the genome that codes for some specific thing. We have lots of genes. Humans have about 20,000. And then, so like genes code for characters, right? Genes affect characters. And hair color would be an example of that. And then within genes, you have specific alleles, and alleles code for specific traits or character states. So in the context of hair color, you might have an allele for the trait of red hair. So I hope it's clear how these different terms are related, because we're going to be using all of them going forward, and though the distinctions are a little bit subtle, it's important that you have the distinction clear in your mind. So now that we've established that background, we can talk about Gregor Mendel. He lived from 1822 to 1884, and you never have to know the years for any of these people that we're going to talk about, but just to kind of give you an idea. A lot of this unit is kind of in a historical context, so it's nice to be able to kind of put things on the timeline relative to each other. The big thing we're going to talk about here isn't just the person, but it's his experimental approach. So he was an Austrian monk, and his claim to fame is that he was the first to really determine the basic rules of inheritance for eukaryotes. Now, I want to be clear, we are talking about eukaryotes here, like plants specifically for Mendel. Prokaryotes, bacteria, they're weird. The basic rules are the same, but the stuff that Mendel figured out doesn't apply to them for a bunch of reasons that we're not going to get into today. So he published his work in the 1860s, and it didn't really receive very much attention at all until it was rediscovered in the early 1900s. And next time, we're actually going to pick up with one of the people in the early 1900s that was working with kind of Mendel's findings and taking the next steps with them. The important thing for today is that Mendel's work forms the foundation of the field of genetics, and Mendel is sometimes called the father of genetics for that reason. So Mendel worked with an experimental organism called the garden pea. Real simple, it's just a pea plant. Now, the pea plant has lots of advantages. It's inexpensive, it's easy to obtain, it's just a common plant. It has lots of traits that are easily identifiable, so like the height, the color of the flowers, the color of the seeds, the texture of the seed pods or the seeds themselves. They're easy to grow and they have a relatively short generation time. I swear I've looked this up a dozen times in my life and I can never remember what the generation time is, so I'm probably going to get it wrong. I want to say it's like 2 months. It could be 3 months. I don't remember off the top of my head. But I know that they have it could be 6 weeks. I know they have a relatively short generation time, which makes it convenient so you could do multiple generations in a year. The other nice thing about the garden pea is that it's easy to control the pollination. So what Mendel actually did is he would go around cutting parts off of the flowers so they didn't self-fertilize. And then he went around with paintbrushes and would manually move pollen between the flowers that he wanted to cross for his experiments. And then the final nice thing about the garden pea is that there are lots of varieties available. Mendel ultimately collected 34 strains that had different traits or different character states, and then he was able to cross them. He was able to make them reproduce in whatever combinations he wanted to determine how those traits were inherited. The other big thing that Mendel did, and this was the really groundbreaking thing, was that he applied quantitative methods to his work. Now, as I said at the beginning, the fact that offspring have the same traits as their parents, that's nothing new. We've known that for thousands of years. But what Mendel did is he used an experimental approach. So he was able to say, okay, I have X and Y, I'm going to cross them, I'm going to predict what I'm going to get. He documented his specific protocols so you can go back and we know exactly what Mendel did, and it's possible today to go back and reproduce exactly the work that he did to check his work. And of course, he recorded data. And this is really important because you can then evaluate the data. Do your data actually match your predictions? Well, if you're not recording your data, you can't do that. The takeaway here is, remember kids, the only difference between screwing around in science is writing it down. And the key thing is that Mendel wrote it all down. So Mendel started by developing what he called true breeding lines for his plants. True breeding just means that they always express the same trait for whatever trait you're focusing on, generation after generation, as long as they're self-fertilizing, no exceptions. In other words, true breeding lines for a particular phenotype always have the same phenotype, whether it's tall plants or green seeds or purple flowers. You can interbreed them as many times as you want. As long as you're staying within that lineage and you're self-fertilizing, then they're never going to change their traits. And in order to use a specific line in his experiments, Mendel first had to generate 2 years of true breeding in order to be confident that they only carried whatever it was that he wanted them to pass on. So we now know that he was isolating specific alleles. He didn't know that. He didn't have any of this vocabulary. But he knew there was something that was controlling those traits. And to make sure that the plants he had only had the thing he wanted, he made sure that they were true breeding for 2 years before he used them in his experiments. So the thing that Mendel did was test a hypothesis called blending inheritance. This is the idea that gametes contain a sampling of fluids from the parents' bodies. During reproduction, specifically during fertilization, those fluids fuse, they blend together, hence the name, and the offspring is therefore going to have traits that are intermediate between those two parents. This was the prevailing idea of how heredity worked as of the mid-1800s. So to test this idea, Mendel set up what are called experimental crosses. We're going to make 2 organisms, and we're going to see what the offspring's phenotype is. And we're going to make predictions about what it should be, and we're going to see if what we expect matches those predictions. And when we do this, we're really looking at 3 generations. The first is the P generation, which is the parental generation. Then we have the F1 generation, or first filial generation. That's from the Latin for son. And then the F2 generation, which you could think of as the grandkids. We really have grandparents, so we have like parents, kids, grandkids is the way you can think about this. So Mendel started by taking 2 different plants, right, plants that have some different phenotypes, and they're true breeding, and he's going to cross them with each other, and that's the P generation. Now we're going to go through and see what else he did and what he found. Now the prediction, because again, we're doing this in an experimental way, we're using the scientific method, so we are hypothesis testing. The prediction is that if blending is accurate, then the F1 phenotypes should be intermediate between the P phenotypes. So if you have, hypothetically, a red flower and a white flower, and the blending hypothesis is correct, then we should see I'm assuming you said it, a pink flower. That's what we would expect in that situation. And that's kind of the baseline for what Mendel was doing going into these experiments. So here's what Mendel did. We're going to start with a true breeding P generation. So in this example, you've got purple flowers and white flowers, and you're going to mate those two true breeding lines with each other. Then you're going to get the F1 generation right here, and you're going to mate members of the F1 generation with each other to get the F2 generation. To get the F2 generation, and then you're going to go through and kind of see what happened. So let's see what happened. Sorry, kind of jumped the gun on that one. So the first thing is that there are 4 observations I'm going to point out here. So the first one is that the members of the F1 generation always resemble just one parent. It's not intermediate, it's not a mix of the two, it's not some purple, some white. They are always representing just one of the parents. The other parent is absent from the F1. Second observation is that the F2, it's not like that. The F2 is a mix of the two P generation phenotypes. So here we have purple in the F1, but then in the F2, we've got purple and we've got white back. So that third thing, the phenotype that was present in the P generation, absent in the P1, it comes back in the F2. And finally, we see a 3-to-1 ratio of those two phenotypes. The one that was exclusively making up the F1 generation, that is about 3/4 of the F2, and the one that was missing makes up the remaining quarter of the F2 generation. That 3-to-1 ratio turns out to be consistent across all these different traits that Mendel looked at. So those are the 4 findings. So to conclude this testing of the blending inheritance hypothesis, no intermediate phenotypes appeared. Lost phenotypes reappeared, which should not be possible if blending was correct. And because the blending of fluids cannot explain either observation, what can we say about the blending hypothesis? We can reject it. Like a ka-tunk sound effect for that, but that's, we can reject that hypothesis. So first thing Mendel did is show that the blending hypothesis is wrong, but you need to have a better answer. So let's see what he actually replaces it with. So part C here is looking at Mendel's model. His hypothesis, the alternative to blending inheritance, is called particulate inheritance. So according to this model, the characters are determined by what Mendel called heritable factors. Each character within an organism is controlled by 2 factors, one from each parent. And Mendel didn't come up with the word gene, but we now know that the heritable factors that Mendel described are in fact genes. That's what this is. So what we're going to do here is we're going to use the word gene going forward. We're going to use the modern language, even though Mendel didn't have that language. But to keep things consistent and minimize the chance for confusion between this lecture and subsequent ones, we're just going to use the modern language. Just be aware that Mendel didn't have the modern language, but we're just going to use it. So there are 4 components of Mendel's model. The first component is the concept of alleles. Again, he didn't have those words, but he came up with the idea of an allele, which is just an alternative version of a gene. So this is something that should look familiar to you since you've talked about meiosis. So we have a tetrad here. We've got 1 homologous chromosome from mom, 1 from dad, and we look over here, we have the gene A. Let's say that's a gene for hair color. It's more complicated than that in humans, but it doesn't matter, just pretend. So we have gene A for hair color. We have an allele right here for black hair, and an allele over here for red hair. We have 2 different variants of that gene. That's the idea of alleles. The second part of Mendel's theory is that there are 2 factors for each character, or 2 copies of each gene in an individual. This is the idea of ploidy, and you've already talked about haploid and diploid organisms, or haploid and diploid genomes. So the idea here is that diploid individuals get 2 copies of each gene, 1 copy from each parent. You get 1 complete set of chromosomes from mom, 1 complete set of chromosomes from dad. That's what Mendel said. Again, he didn't have the word chromosomes, didn't know what they were, didn't know what they did. Just that's the modern way of saying what Mendel figured out. Basically, he figured out for his pea plants, and kind of coincidentally, also for us, we're diploid. We have one copy from each parent. Now, these copies could be identical, which is the case in true breeding plants, or they may be different, right? So you could have here, they're different, but they could also be capital A on for both parents. That works too. And as you probably know, these genes that are the same, right? You have the same gene, one from each parent. Those are found on homologous chromosomes. So chromosome number 5 or whatever, it's going to have the hair color gene right here from mom, and then dad's chromosome 5 going to have the same hair color gene in the same place. You could have different alleles, but the genes are going to be in the same place on the same homologous chromosome. The third part of Mendel's model is dominance. So if 2 alleles differ, the dominant allele is the one that determines the phenotype, and the recessive allele has no noticeable effect. In other words, the dominant allele covers up the recessive allele when it comes to phenotype. I will say right up front, it's more complicated than this. There are lots of traits that don't follow this, but the traits Mendel looked at were simple dominant recessive traits, which makes it easy. So here's an example for flower color. In the pea plants that Mendel studied, purple, capital P, because capital letters tend to represent, or I should say typically represent dominant, and lowercase represents recessive, but you keep the letter the same, and the letter is based on the dominant. So in the pea plants for flower color, purple, capital P, is dominant over white, lowercase p. That's the example we're going to use. So if you have 2 copies of the dominant allele, and we'll get to what that's called in a minute. If you have 2 copies of the dominant allele, that's a purple flower, because you only have the information to make purple. If you have 2 copies of the recessive allele, it's going to be white because that's the only option you have. If you have 1 purple allele and 1 white allele, since purple is dominant over white, then the phenotype matches the dominant allele, and those flowers will be purple. That's what we mean by dominance. One allele can cover up the other one when they're both present together, and that is called dominance. The dominant allele covers the recessive allele. And finally, the fourth part of Mendel's model are the 2 principles of heredity, sometimes called Mendel's laws. These are the law of segregation and the law of independent assortment. So let's go through what those mean. The law of segregation just says that the 2 alleles for a character segregate or separate during gamete formation. Each gamete only gets 1. Now remember, Mendel didn't know anything about meiosis. He didn't know anything about chromosomes carrying genes, none of that stuff. But just by following the traits through generations, he was able to kind of figure out what was going on here. So you've got, dad's genes for hair color, dad's alleles, I should say, for hair color right here, mom's right there. And during meiosis, they are going to separate from each other. So the gametes only get either dad's or mom's, but not both. In other words, the law of segregation describes the reduction in ploidy from diploid to haploid that occurs during meiosis. Now, just a quick review. What stage of meiosis is this? Right, they're lined up and then they separate. That is, it's anaphase. Remember, what do you need? Anaphase 1. And that matches what you've learned about meiosis. Because when does the reduction in ploidy occur? It occurs during meiosis 1. Well, segregation, the separation going from your connected tetrad with moms and dads, separating that into one or the other into the daughter cells, that happens during meiosis 1, specifically anaphase 1. So that actually matches what you've already learned about meiosis. So we can do an example of this. We can look at seed color in the pea plants. Yellow, capital Y is dominant over green, lowercase y. So your possible genotypes in an individual are homozygous dominant. That's when you have 2 of the same allele for a particular gene or locus. Locus just means location in the genome. So you could be capital Y, that's homozygous dominant. You can be homozygous recessive, 2 alleles for green, or you can be Heterozygous, this is when an individual has two different alleles at a specific locus. So you have one dominant capital Y allele and one recessive lowercase y allele. So the possible gametes in an individual for seed color. What gametes can you get? Now remember, gametes have to be what ploidy? They have to be haploid. So if you're going to make haploid gametes, you only get one of these alleles. So you can get either a dominant allele or a recessive allele. Those are your options for gametes. Why is that the case? Because they segregate from each other during meiosis. So now we can talk about the second law, the law of independent assortment. This states that genes on different chromosomes assort independently during gamete formation. And this is due to the random, I will say, Mendel did not know anything about chromosomes. He just said the heritable factors for different traits assort or for different characters assort independently during gamete formation. He didn't say anything about chromosomes. And next time we'll see why he's a little bit wrong about this, but we will use the modern language the way we know that this law is correct. The correct way to state this is that genes on different chromosomes assort independently, and that's due to the random orientation of tetrads during metaphase 1. Next time when we talk about chromosomes, we'll see how Mendel didn't know anything about chromosomes. He thought everything was independently assorting from each other. We know that's not the case. It's based on the chromosomes, so just keep that in mind here. Mendel wasn't quite perfectly right about this, but if you say it applies to chromosomes rather than genes, then you're good. So the thing here is that during meiosis, specifically during meiosis 1, these different chromosomal orientations are equally likely. So you could have your chromosomes oriented, your tetrads, I should say, oriented like this, right? You have them oriented like that. You do it like that. You do it like that, right? One chromosome, one tetrad, second tetrad. It's random. So this one can be like this, and moms go one way and dads go the other way. This one, moms go the same way or the other way, or you could flip it. It's random. So these different conformations are all equally likely. And this leads to new combinations of genes among the different chromosomes. This is one of the major mechanisms of genetic recombination. You've already talked about another one, right? Crossing over. So you've talked about crossing over during meiosis. And now we're talking about independent assortment during meiosis. They have the same effect, which is generate new combinations of alleles in the offspring. And as we'll talk about down the road, this is basically how sexual reproduction leads to more genetic diversity, which is like the important thing about sexual reproduction. We'll talk about it more as we go. Now keep in mind, Mendel didn't know the mechanisms behind any of this. Nothing about DNA, genes, chromosomes, like none of that stuff. He didn't know what was going on. He's just describing what's happening. So we can look at this kind of graphically here. Each of these pairs right here represents a tetrad, or a pair of homologous chromosomes. Now, you could just kind of visualize a genome with 8 chromosomes, right? 4 from mom, 4 from dad, all right? And arranged like this, there's new combinations. You're either going to get dads or you're going to get moms. But we have 2 mechanisms that we know of now that mix and match. So we just introduced independent assortment, where the orientation of the tetrads changes, which means you're going to get some of mom's chromosomes going one way, and then in that same direction, you're going to get some of dad's chromosomes. And we know that within the tetrad, you have crossing over. We already talked about that. So within the chromosome, you can mix and match. Between the chromosomes, you can mix and match. And the result is that in the gametes, instead of getting all of mom's alleles, or I should say all the alleles you inherited from mom, or all the alleles you inherited from dad, all of your gametes have a mix of alleles inherited from mom and dad. And this generates new combinations, which means we're generating new diversity within that population. And that's the importance of these 2 processes working in concert.